Thank you. Okay, welcome everyone. We are going to go ahead and get started. So Dr. Huppert has all the time. Very excited about this session. And if you are sticking around for later this evening, we have a very special recognition of Dr. Huppert at our Champion Award celebration tonight. So I am going to let you just go ahead and take over, Stacey.
All right, thank you.
All right.
Hopefully this works the second time. Did it work? Full screen?
Looks good.
All right. So thank you very much for allowing me to share some of our research today. You'll see that the title is different. I think we had so many emails going back and forth that I think something got dropped somewhere along the line. But this is amazing, and I'm so glad that you guys were able to pull together a virtual meeting this year since we didn't get to do in person. I think it's really important for the community to stay connected. So I'm really going to go over 3 vignettes, stories. If we don't have time, that's great. I'll kind of watch the time. I want to make sure that we have time for a discussion. I'll stay kind of high level, but still give some scientific details for all the scientific guys out there in the audience to maybe have a little bit of meat to dig your teeth into. Oops, and I need to use this. Okay, but I do want to start off. So this is a picture of my group at Cincinnati Children's. And you can see the group, we have our Alagille Aware Day t-shirts on. Most of the work I'll be showing you is from Ashley Cast, John Barwinkle, Sanjay did some of the single cell sequencing, and then my sister, Carrie Huppert. So Emma did a great job, and so I may be able to shorten some of these slides, but I just wanted you all to get a picture of how intricate the intrahepatic bile duct system is. And the idea that even though I'm going to show you some defects in that in our Alagille mouse models, that what we're trying to do to rebuild that, you can see how complicated that structure is. It's very hierarchical. It's pretty systematic in the way that it's built. It has a pretty regular architecture. And as Emma pointed out, that this architecture follows the portal vein. And now that all of that is connected intrahepatically, of course, has to connect extrahepatically for all of the bile to drain out of the liver and into the duodenum. You can see this fine little yellow tube here. So my group has really focused both on development and on regeneration and injury. And as Dr. Piccoli pointed out yesterday, there really hasn't been any progress in bile duct augmentation. Even though we've known this pathway for a while, as the group at CHOP and Nancy Spinner identified that the NOTCH pathway is involved, we really still haven't gotten to the point of being able to augment bile duct structure. In development, I don't think this has been discussed, but there is a liver progenitor cell, and it's bipotential, which means it has the ability to give rise to both hepatocytes, the main parenchymal cell of the liver that does metabolic work and secretes the bile, and cholangiocytes, which are, of course, the cell that contributes and builds the bile duct system. And I'm going to show you just a little snippet of our original work showing that in the adult liver, there is this ability in a normal liver in order for cells to be very plastic and that they can transdifferentiate or go through a dedifferentiation and proliferative process to give rise to each other, which makes the liver an amazing organ in its ability to regenerate. Of course, in times where there are genetic mutations and other diseases that are chronic, it lacks the capacity to do this. So just to give you an idea, the liver is formed from the ventral foregut endoderm, and it's very different from the other organs that are formed from that ventral foregut endoderm, the lung, the thyroid, are different in the fact that they have an epithelium that branches and grows as the organ grows and takes up space. The liver is different in the aspect of once it gets defined as its progenitor cell, the hepatoblast that's down here, by 13.5 days, the liver in the mouse is the size of a 9.5-day embryo, and it still doesn't have its bile duct system built yet. It's only after this stage that it starts to build that bile duct tree, that epithelium, whereas these other organs build it as it grows. So it's different. And Emma went through this intricate dance that the cells, as they begin to be specified to develop the bile duct from cholangiocytes, that they're specified around the portal vein and only around the portal vein. And you don't need to get into the complicated genes and nomenclature here, but just to see that there's this morphogenesis that happens, that the cells are moving around, finding each other and making tubes in 2-dimension, that of course in 3-dimension are connected to the outside of the liver. And this is just showing you one of our ink injections that Emma had showed, where you have the portal vein that is filled with a white ink and the bile duct system that's filled with a black ink. And you can see that there is this a bigger caliber duct that's running along. And then you see these really fine tubes that are also connected in there. So we need to think about this system developing in 3-dimension. It's been published for a really long time from Frederick Lemegre's group that the system forms from the hilar, or the outside, connecting to the extrahepatic bile duct, going inward to the periphery of the liver, or the outer edge of the liver lobes, where it's specifying cells and they quickly remodel into tubes. And here's showing you an ink injection from Naoki Tanamazu's group. You can see this very fine homogeneous architecture that's surrounding the portal vein at embryonic day 17 that Emma was telling you, our mouse nomenclature. And once bile is starting to be produced by hepatocytes, you can see a change in the remodeling from embryonic day 17 to 18, where you now see there is a tube that's a higher caliber, and you still see finer network out here in the periphery, which is diagrammed up here. And then postnatally, you can see that now you have even more branching of the system that have a nice hierarchical structure, going from larger to smaller to very small, that have this homogeneous network. So there's this big change that happens 3-dimensionally, and it seems to be coincident with bioproduction. So here are my 3 vignettes that I'm going to try to get through. The first, if you've been at a meeting in the last couple of years, you've seen Holger present some of our work, and I just want to give that at a high level regarding hepatocyte plasticity and the potential for hepatocyte to cholangiocyte transdifferentiation and de novo bile duct formation. So this is a potential way for the liver to regenerate. The second is going into a new area, which is niche requirements for cholangiocyte specification in intrahepatic bile duct formation. And we're collaborating with Robert Schwartz at Cornell Medical School and Takanori Takebe here at Cincinnati Children's. And then the third is genetic modifiers as potential therapeutic targets. And we have preclinical studies going on with Hamid at Baylor College of Medicine. And you heard Hamid's name earlier from Emmett's talk, that has the heterozygous jagged mouse that is just like patients that have haploinsufficiency of 1 copy of jagged that's mutated. And then also our Cincinnati Pediatric Cell Atlas Center with Nathan Salmos and Takanori Takebe, sequencing liver at all different ages of development in order to build a map to then be able to go back and overlay diseased liver on top of that to understand what's going wrong. So initially when we got into looking at bile duct regeneration, most of the models that were built were damage models. So feeding a mouse different types of toxins or performing surgical procedures on them to block bile drainage. And in all of these cases, those mice start with a normal bile duct architecture. And when they're given those toxins or a surgical procedure to block the ability of bile to flow out of the liver, is that they increase bile duct mass. So that's not Alagille syndrome. And so we needed to build a different model. We needed to build a model, as you heard yesterday in talks from David or Dr. Piccoli, is a paucity of the bile ducts. So we want something that doesn't have the full structure, and understand if there's some sort of selectivity that you could get regeneration and rebuilding of the bile duct system. So to do that, as you all know, as you're here at this meeting, we initially used mice in the NOTCH pathway, and we built a different allelic series, basically inhibiting the ability of the pathway to single greater and greater levels. So you've heard about the jagged heterozygous mouse. We also had, there are 4 NOTCH receptors. We deleted 2 of the receptors that are expressed in the epithelial cells that might be potentially involved in bile duct development. And then we finally deleted RBP that you see down here in this left corner. And these are resin casts of the bile duct system. Here is a control or normal bile duct system. And then here is one in an RBP knockout mouse. And this is the DNA binding partner required for all NOTCH receptors to signal and to turn on genes. So this is the business end of the pathway. The goal of the pathway is to activate from ligand to receptor, and then to bind to DNA and activate gene transcription. We also have shown that if you just use this active component, that you can increase and get way too many bile ducts to form, but they're all communicating and connected to the outside extrahepatic bile duct. But so we wanted to build a model that was deficient and understand if we could rebuild a ductal system from that. So even though this looks bad compared to this, these mice aren't that sick. So we decided to look at what was known in the molecular pathways required for cholangiocyte bile duct development. And the other molecule that we went after was HNF6. And this is involved both interacting here with the NOTCH pathway to drive SOX9, but also interacts with the TGF-beta pathway. And it really is YAP, NOTCH, and TGF-beta are the main drivers that are known thus far to really require to get cholangiocyte activation. So we knocked out both of those pathways using RBP and HNF6. So now, as you can see on the right side of your screen, we now have a very extreme model that has bile duct paucity. By an Inca injection, you can see that they really only put in a couple trunks of the tree, and then there's nothing peripherally that is built, and the animal is very jaundiced. So this does provide us this model that we can then begin to study the ability of different cells and in vivo reprogramming to regenerate the bile duct system. What was a surprise to us is that there were improvements, and Emma alluded to this with their jagged nodder mouse, that there seems to be this improvement over time. And I do want to point out that all of our studies are done with the left lobe in the mouse. It's the most consistent, and it is the most severe phenotype of all the lobes in the mouse. You can see that the control or normal, the bile duct system at 30 days after birth and at 120 days. And then here's showing you at 5 months, and you can see this beautiful hierarchical structure that you have larger caliber that feeds into intermediate size, and then smaller, and then finally really, really fine structures. With this double knockout model that we made with RBP and HNF6 deleted just in the hepatic epithelial cells, you get a very truncated that I showed you earlier, but then they go ahead and remodel this ductal structure. It is tortuous, so it's not completely normal. It doesn't, and you can see that these ducts kind of wind around. There's not a beautiful hierarchical structure, but the fibrosis and serum chemistry come back to normal. I should note for all the scientists out there that those ducts that are built have not escaped deletion, so they still are deleted for RBP and HNF6, and that those ducts are stable. So once you resolve the injury, they don't go back to truncated, they remain there in place. So then we got involved with Holger Willenbring's group. So he's done a lot of lineage tracing using different viral vectors to infect specific cells to allow us to modulate genetically in a transient way. And the reason we wanted to do this was to understand when those ducts are being built de novo, are they coming from hepatocytes? And scientifically, it was very difficult because we used some genetic tricks to knock out these two genes with one way of forming deletion. And so we needed to come up with another way to form deletions so that we could label these hepatocytes. So we infect the mice with this virus that has basically a flippase or an enzyme that allows us to recombine a reporter, and that will label all the hepatocytes green. The hypothesis is if those extra ducts that are being built are coming from hepatocytes, they will now have both the red label that's for cholangiocytes and the green label that came from hepatocytes, and green and red make yellow. And that's what I'm showing you here. This is showing you the portal vein that Emma had in some of her slides, and these bile ducts here that are labeled with both a hepatocyte marker, and you can see the hepatocytes all labeled out here, and the cholangiocyte marker that's labeled in red, wide spectrum keratin. So those new ducts that are being built from hepatocytes. And this is just showing you, as we expected since cholestasis was resolved, is that all the cholangiocytes that are making the bile duct structure are labeled with this red marker. And only the hilar, or those main truncated ducts that were there originally, that are labeled with this lectin in blue. And then all the hepatocyte-derived ones are green. So you can see in this video that those hepatocyte-derived cholangiocytes that are composing this duct are coming in and finding that remnant of a trunk that was there during development. So they find each other in space and they build each other out into a duct. So this first conclusion is that hepatocyte plasticity and transdifferentiation has extensive tissue repair capability, and it has the potential to build the intrahepatic bile duct system and resolve the cholestasis and related injuries. So now I want to turn to a different model to really try to understand the niche requirements for cholangiocyte specification and intrahepatic bile duct formation. This really gets to the question of, if hepatocytes have the ability to do this, why don't they do it in Alagille syndrome? So first I want to give you a few key observations that highlight that this does not happen in an efficient way in Alagille syndrome. So the first observation is this study that was part of the Gala group, again with Benita Kemath and the Children Network, is that children that have a liver defect with Alagille syndrome, by the time they reach young adulthood, around 19 years of age, that less than 25% of them have their native liver, meaning that 75% of them have had a transplant. So this suggests that hepatocyte transdifferentiation is not efficiently happening in Alagille syndrome. I also want to point out, and this again is the jagged heterozygous mouse, so this is just like patients with haploinsufficiency, that they make a very truncated bile duct system. This is at a month at age. At 4 months of age, you can see that that system has started to remodel. You get more of it. But you can see that even at 7 months of age in the periphery, there's still this homogeneous network that, I don't know if you remember, but when I showed you that embryonic structure, it looks very embryonic. So it's not able to go ahead and form this beautiful hierarchical structure that goes all the way to the periphery of the liver. We've also been doing a lot of single cell RNA sequencing, and this tells us in each cell of the liver what the expression pattern is for all the transcripts. And I'm showing you on the left are all the cells from 2 different biological liver samples that are wild type or normal. And on the right are 2 jagged biologicals. And I hope you can see, I forgot to put my circles on here this morning. If you look at this epithelial cell population, that's this turquoise color that we call jagged C22. And it's because it's basically only in the jagged C22. So these cells have both expression of hepatocyte markers and cholangiocyte markers, which you can see down here in C8. But in wild type, there's clear hepatocytes and there's clear cholangiocytes. In jagged, these populations are running together. So the cells are there, they just don't ever fully commit to the cholangiocyte compartment. So what's missing? The other interesting population that's we see that's different is this macrophage population here. And we're starting to work on that, but I don't have anything to report today and whether or not that population is highly involved in remodeling the liver and it's causing a problem with that in jagged. The other thing that I want to point out is that in this double knockout model that was so amazing in its ability to regenerate the bile duct system was that we only knocked out the Notch pathway in the epithelial cells. So the endothelial cells that Emma was talking about and the mesenchymal cells or the smooth muscle cells that Emma was talking about are all wild type. So in other words, you don't need reception of the pathway in the epithelium to get the bile ducts to remodel, but there might be something wrong again with the niche. And the last, I think this is the last observation is that with Holger's group again, we did transplants both with mouse at different ages, hepatocytes isolated from different ages, different genetic backgrounds, but we also took human hepatocytes, isolated, transplanted them into the mouse, and looked to see if they gave rise. So is this a mouse-specific phenomenon, or can they also happen in human? And what we saw was this human nuclear antigen is green, and bile ducts are marked with red, and you can see that those hepatocytes that we put in have transdifferentiated into cholangiocytes. You can see that they really don't go into the hepatocyte compartment. When they got into that environment, they sensed that there was a problem with bile ducts, not a problem with hepatocytes. And so they immediately changed their identity and contributed to the bile duct system. So it's a signal that must be missing in a jagged heterozygous mouse and potentially the patient. So what's required? I think this paper was also mentioned earlier. So this is Luisa Aurelia Raspe. She's really a vascular biologist, but she's also been in and out of the Notch field a lot. And if you are at the scientific meeting in 2022, she will be one of the speakers. She has some new exciting vascular data, but she did this study to really understand where jagged was required. She used SM22 to delete jagged only in the SM22 or smooth muscle cells. And she used BE cadherin to understand and delete jagged in the endothelial cells. And as you can see by her pictorial right here was that it really is those smooth muscle cells that require jagged. So if you knock it out in the endothelium, the mice and the bile duct structure was completely fine. And what Emma was showing earlier in her studies is that if you are with her not her mouse, but here is just knocking it out in those smooth muscle fibroblasts, you can see that you don't get the cells, there are cells there, but they're not remodeling properly. They don't make a nice duct. So I really wanted to find a molecule that would mark that niche that's there around the bile duct. And I wanted, importantly, which I wrote here, a signaling pathway so we could get at mechanism that we could find targets or therapeutic targets, so we could start to think about trying to modulate that niche. And so this is a paper by Robert Schwartz, and he was looking at injury and looking at fibrosis. And what he noticed was a marker in the hedgehog pathway, one of the downstream components, GLI1, is surrounding the bile ducts and tightly surrounding the bile ducts. You can see a section through a mouse liver here that shows the bile ducts here in green, and those mesenchymal cells or smooth muscle cells marked with GLI1. And you can see this is a hilar duct. This is getting out there at a tiny peripheral duct. And you'll notice that as you get out into the canal herring, which is really the connection with hepatocytes and cholangiocytes for bile to drain, that they don't seem to be surrounded by GLI1 positive cells. What is an interesting observation that he had, and Emma mentioned these reactive ductules, is those reactive ductules are surrounded by stellate cells. So he did different labeling and showed that it's really something that maintains the bile duct phenotype and is a crucial bile duct is surrounded by GLI cells. But the things that are remodeling and reactive are surrounded by stellate cells. So I don't want to get into too many nuances, but I know the scientists will want to know the cellular source and which hedgehog ligand. So I just want to show you work from Robert's group again, showing you that the cholangiocytes, both extrahepatically and intrahepatically, express Indian hedgehog. So it's not sonic hedgehog, which might be expected. It is Indian hedgehog. So we've been using the GLI1 mouse to really understand three-dimensionally where those cells are in the bile duct architecture. And I hope you can appreciate in this image that those cells, which are labeled with blue now, are tightly associated with a bile duct. In fact, it almost looks like an ink injection, that they're tightly associated. You can see both the central vein, which doesn't have a bile duct here, and the portal vein that has the bile duct wrapping around it. If you section through that, you can see the blue GLI1 positive cells surrounding the bile duct here. Here's the portal vein, here's another bile duct, and those GLI positive cells are surrounding them. Looking at three-dimensionally, you see, and these are 2-month-old mice. Lexi, again, is showing you where the GLI1 cells are, and we're using a marker, osteopontin in green, to mark the cholangiocytes. You can see a bile duct tube coming out at you three-dimensionally. Here's the portal vein, and here's one of those tiny little tubes that sits and surrounds the portal vein. And you can see those GLI1 positive cells around the intrahepatic bile duct. And then this is a 7 or 8 old mouse, retired breeder, and you can see, I think in this high mag, it's really nice because you can see those, kind of get an idea that those are single cells that are blue labeled that are just running around that bile duct along the portal vein here. Oops, I froze, okay. So this is getting a little bit deeper and showing you kind of a wide view, stepping back, where we're looking at a main trunk of a portal vein, the bile duct, and these GLI1 positive cells that are wrapping and even going out towards the intermediate and periphery through, and that's 2 months of age. And here is a week of age. We can see that things haven't completely resolved. There's denser packing of that fine network along, as you can see in the schematic, and here is Tanamazu's ink injection showing the hierarchy, and then out here in the periphery, you can see this denser network. So you can see the bile ducts are in red, and those mesenchymal cells here are in white. And I hope you can see over here, it's a little bit clearer that they have chosen a more larger trunk here, and then there are these fine networks. So I wouldn't be telling you this if it wasn't conserved, because if it's not conserved from mouse to human, there's no reason to try to figure it all out. So this is RNA-scope looking at the transcript of Indian hedgehog in a human section of a human liver. And you can see those red dots of Indian hedgehog in the bile duct. And then you can also see the glee red dots surrounding a bile duct. And here's just showing you an immunostain with the bile ducts are marked with this purple with cytokeratin 19, and then PDGF receptor alpha, which we see some of the glee cells overlap with. And so to do it by protein, you can see that they're tightly associated and wrapping around the bile ducts. So is the hedgehog pathway involved in specification, and when are these GLE1 positive cells first observed in the liver? So trying to do a temporal and spatial. So we already knew from studies from Pam Hoodless's group in Canada at Fox Laboratory, and from Aaron Zorn and Taka and Jim Wells' group here at Cincinnati Children's, looking at single cell sequencing early in the mouse embryo. So looking from E7.5 to E9.5, E10.5. And so this is a lot to show that this diagram here on the right, which says that hedgehog, these positive arrows are in the gut tube and also in the lung bud, but the pharynx mesenchyme and the liver mesenchyme is off. So up to the point of 9.5, 10.5, the liver doesn't have hedgehog GLE1 positive in it. Kentaro Iwasawa is going to give you a talk more about these extrahepatic ducts and forming boundaries in one of the science spotlights later. But I just wanted to give an idea. At E11.5, now you start to have a structure where you have a gallbladder and everything that's connecting to the liver extrahepatically. But the liver is over here, and you can see with PDX is marking that, but it's basically, I'm going to show you in a slide, it's negative still. So we looked at 14.5. Even then, at that point in time, you can see as described by the earlier single cell, that the lung is highly hedgehog positive, GLE1 positive, the stomach and the duodenum and the gallbladder and extrahepatic are positive, but the liver is negative. It's not until about 15.5, which it doesn't look very organized, and 16.5, I'm going to show you a slide where you can see a bunch of different embryos that we looked at to show that now you start to see it going into the liver. I should have showed this slide first. Now it's a little bit 13.5, 15 just starting, and 16, you can see those portal veins coming into the liver and surrounded by these GLE1 positive cells. And this is just showing you it's not a random N of 1. Here's showing you 4 embryos, but I think we had 6 embryos at embryonic day 16. You can see these nice structures of vessels that are going in that are have GLE1 positive cells surrounding them. So does this match with intrahepatic bile duct development? So at embryonic day 15, this is a multiplex RNA scope. SOX9 is one of the first markers for bile ducts or cholangiocytes to being specified. Jagged, we already know, is important in those smooth muscle cells, and then GLE. And you can see here, the SOX9 positive cells are on the outside, and you can see the jagged and GLE expressing cells are surrounding tightly the portal vein. By immunofluorescence and looking at protein, again, you see SOX9 cells, and they're surrounding these PDGF receptor alpha, which is green, and the GLE marked by LAC-C here, that's close to the portal vein and surrounding it. So I wouldn't be telling you this if it didn't seem like it was important in a jagged haploinsufficiency. So these are showing you some of our first inroads into looking in the jagged heterozygous mouse. So again, the haploinsufficiency like Alagille patients. And looking with GLE-LAC-C so we can trait or look at which cells are expressing and if they're still there. So this is a normal animal here on the left. And you can see GLE-LAC-C cells surrounding the bile ducts. You can see a less severe phenotype. You've heard that a lot, and you know with patients that there is this variable phenotype. So you can see this one has a truncated bile duct system, but there is some bile duct there, and it's surrounded by Gli1 positive cells. You can also see that it seems like there's also Gli1 positive cells that are still surrounding the portal vein. It's like they didn't receive the cue to remodel. And then in a severe phenotype that has no bile ducts, you can see the Gli1 positive cells are basically surrounding the vessels and haven't remodeled or found a bile duct to remodel around. This is just showing you a section through these livers after they were cleared and stained. And you can see that the cholangiocytes are specified, but they haven't remodeled into nice ducts, and they're not fully surrounded by these Gli1 positive cells. So they don't have the support mechanism and potentially this correct signaling to go ahead and remodel. Even at later time points, so these are 2 months of age. Now I'm showing you the ink injection at the top, and you can see how the caliber gets larger and larger as you go out to the periphery. You can see that in a jagged heterozygous mouse, there's some that are really large and then they get fine, and there's not a consistent going from larger to smaller to the edge. And then this is a really severe model. You can see here just a few jagged or a few Gli1 cells surrounding those bile ducts. And here you can see that there really isn't a bile duct here per se in this region. And those cells are kind of just sitting there not knowing what to do. So we know by exposure levels that in a jagged heterozygous mouse, I figured out my red circle here, that you can see the LAC-C level is lower, so the white isn't as intense. And you can see these nice ducts and then some unremodeled things that are here in the jagged het. But now we're to the point of understanding, is this due to a cholangiocyte problem, a mesenchymal identity problem, or both? And this is just to show you from a kiddo here at Cincinnati Children's that had a transplant. And you can see that this duct that's really just kind of, we had to search to find one, but here's one kind of by the marker that's purple for cytokeratin. And what's really interesting, I hope you can see, is that they now do not have PDGF receptor alpha cells surrounding them. These reactive cells do, that are kind of out here, but they're not part of the main connected draining system. So this second part, niche requirements for cholangiocyte specification, again, we're just diving into this. We have a bunch of other necessity and autonomy studies going on, but the niche is impacted by jagged haploinsufficiency. And I just said this, we're testing a primary defect and necessity insufficiency. And we're building with TACA some organoid models to really look at sufficiency. Can we isolate the different mesenchymal? I only showed you the one population, but we now know there's about 3 populations with overlap of signals, and putting those into the organoid models to try to figure out sufficiency. And then the last topic quickly I want to go through is genetic modifiers. And basically, we can't share a lot, but next year Hamid is one of the speakers. And so by that time, we will be able to share quite a bit. Sorry. So this is Hamid's group at Baylor College of Medicine. Josh Adams was one of the main players. He's now in his residency. Nima has also been a main player. And then he has a new graduate student who's not pictured here, is Fox Duncan, and I think, or Duncan Fox, and I think that we will see some great things from him. But this is the paper that Nancy had showed yesterday, Dr. Spinner, where Hamid and I were looking at the modifier of SOX9. And I don't want to belabor too complicated genetics here, but they're basically in this order of this is normal, this is still normal. So if you just heterozygous of SOX9, it's still pretty normal. Heterozygous of jagged, as we all know here, causes truncation. And now if you have the heterozygous jagged and heterozygous SOX9, you can see this is the least severe that we had, and this is the most severe. So you basically are getting even more truncation with the deletion of SOX9, and we had many other assays in this paper, or no ducts formed at all. So we are now, we've worked with Ionis to screen for antisense oligos that can be, for our mouse studies, we're using them as just as a sub-Q injection, which is really easy, but we're looking for targets for antisense oligos that increase SOX9 level, hoping to augment bile duct architecture. The project that we're really far along on is this project, which Hamid had published a few years ago with deletion of one copy of PO-glue or Rumi, that he was able to improve bile duct architecture. So PO-glue is a glycosyl transferase, and it's responsible for O-link glycans to epidermal growth fact-like repeats. And if in the protein structure of jagged and NOTCH receptor, they contain EGF repeats, so they're targeted by PO-glue. I think we'll need to mute, thank you. And I should tell you by RNA scope, we have confirmed that PO-glue is expressed both in the SM22 and GLI1 positive cells. And of course, there's some single and double different populations, hepatocytes and cholangiocytes. So the goal is to decrease PO-glue level to augment bile duct structure. I can say that we have very promising results in the liver. And now we're looking at other organs to see if we have also improved the vascular phenotypes in them. And what's nice about Ionis's antisense oligos is that they have non-selective ones, which mean they can be uptaken by any cell type, and also selective chemistries. So they're taken up, for example, by just hepatocytes. So we'll be able to target specific cell types or see if a non-selective one, thus far, it doesn't seem toxic at all. And so maybe that's the way to go, that we can actually improve the ability of jagged and notch signaling in all cell types. So this is just showing you the single knockout bile ducts per portal vein, which Dr. Piccoli talked about as a parameter of paucity. So the single knockout, we know that it's reduced from wild type. We know if you reduce both jagged and Rumi, or what's PO-glute here, that you improve that. And that's just heterozygous, so it's not hitting all the cells, only 50% reduction. And then there's no statistical difference between knocking out Rumi by itself. So we're really excited about these results, and we have, as Sharon and Roberta know, we've been submitting a preclinical grant to NIH, and hopefully this last one we just submitted will be funded. But in order, as I think has been said multiple times from Emma and also Dr. Piccoli and Dr. Spinner, as science evolves, we're able to get newer techniques and higher resolution in order to find defects or to assay. And so it's been really important in going into preclinical studies that my group with Hamid has been working on, trying to come up with really fine assays to really assess the phenotype and whether we're improving the ductal system. So these are showing you really deep images into the bile duct structure. This is wild type, and you can see that duct here stained with osteopontin, and then those fine branches that are coming off of it. This is one huge portal vein here. This is intermediately into the lobe, and you can see now a bile duct, and then some of these fine branches that are wrapping a portal vein structure. And then periphery, this is at postnatal day 17. You can see they're kind of starting to choose a main duct, but they're still pretty much close to a homogeneous architecture. In the jagged haploinsufficient model, this looks very different. So you can see that the cells are there, but they have this very fine homogeneous architecture. And you can even see, as we get into higher magnification, that there are missing cells. They basically don't make the homogeneous structure accurately. And then here's the periphery where that is completely not remodeling properly. So I also liked Dr. Feinstein's talk yesterday. Fine architecture is very important to assess phenotypes. And the last is our single cell data. And I hope, okay, perfect. Is our single cell data. And so we have been building for a couple of years now at Cincinnati, a pediatric cell atlas. We focused on 4 different organs, initially kidney, liver, which is led by myself and Takanori Takebe, bone marrow and intestine. And not only are we doing single cell, but we're also doing very deep imaging. You can see over to the right where we worked out multiplex RNA scope to look at transcripts that we see in single cells to see where they are located architecturally. So my group has been heavily involved in everything that we're doing is being put out publicly as far as protocols for people to use. And this is just showing you, because someone asked yesterday about epigenetics. So we have worked out the protocols. This is just showing you 2 different donor livers. And then an explant, this is chronic rejection, not Alagille. The Alagille livers just came off the sequencer this week. But we're doing both RNA sequencing to find the transcriptome in each single cell of the liver. And because it's multi-omi, we're also getting the same ATAC-seq, which tells you the chromatin structure within the liver that matches with the RNA-seq. So we're gonna get a lot of fine detailed information from these analyses. So I think that's it as far as the genetic modifiers. I think we're really excited, and things are moving really well for some of these collaborations and antisense oligos that we have from Ionis, and we will definitely update you on all of that next year. And then I have been very privileged over the last few years to work with the Alagille Alliance. Thank you, Roberta and Cher, to organize the 2019 meeting, right? Because we were supposed to do 2019 meeting. And then I have the privilege of working with Kathy Looms this year for the 2022 meeting. And hopefully, ladies, I have the right date there. I think that is right. Yeah. And Kathy and I will, after this is off your plate, we have to talk soon because the grant goes into NIH. So I have to get some budget stuff from you guys soon. But anyway, I'll love to take questions. Thank you.
Great. Thank you so much, Stacy. Anyone? Oh, okay. Let's see, Emma, go right ahead. Jump right in.
Sorry, I was clapping, but I actually do have a question. Sorry, I really love the regeneration story, and at least I've had more time to think about that one because that's like a Bible for our lab, that paper. But all the other work was also really, really impressive and exciting, and I'm looking forward to reading all those papers. For the regeneration story with the transdifferentiation of the hepatocytes into cholangiocytes, could you give a little bit more rationale for knocking out HNF6 as well as RBPJ? Because for me, that's always a bit of a mystery why it works to have a jagged 1 heterozygous mouse, but not to just knock out RBPJ.
Yeah, so initially, this is a great question. So initially, we do everything in a knockout necessity model and then a constitutive activation model. And I haven't shown any of that data, I don't think, with the constitutive activation, but it kind of gave us a hint initially that deleting HNF6 helped you produce even more cholangiocytes. So I was really interested what would happen in a loss function if we knocked it out. And I was surprised by there being fewer ducts, but then they regenerated quicker. So the thought was maybe it was helping to increase genetically in the genetic background, increasing TGF-beta activity. If all the models are correct that HNF6 inhibits TGF-beta, if we take that out of the picture, now you've increased TGF-beta. It doesn't help me explain the initial phenotype being worse. But as far as the recovery, I think it's a very much a positive. So if you put HNF6 loss in the background of activated NOTCH, the whole liver will go SOX9 positive.
so does that mean that the recovery is slower or not seen at all if you just have the RBPJ knockout and you don't touch HNF6?
So initially we thought it was because the cholestasis wasn't as high even as in a jagged het. And I think that still might be the case. So the injury level and selection in the liver, it's not reading out as a really severe model by itself.
Mm-hmm.
Because we can put in wild type hepatocytes and they transition. So you don't have to have HNF6 loss to do it.
Okay. Do you think that means that jagged 1 is doing something that is RBPJ independent if a jagged 1 loss is more severe than an RBPJ loss?
No, I think it's because jagged has a role in more than one cell compartment. That model has the niche is completely wild type in the double knockout.
Mm. Okay, thanks. I'll stop monopolizing the conversation.
That's okay.
But fantastic talk.
Thanks.
Duke, welcome. Yeah, Duke, welcome to go ahead.
Hi, nice talk, Stacy. I have a more technical question because I'm not familiar with the reagents, but with the ink injection, you get this really beautiful branching. And then you showed at the end the oxyopathin staining, which also shows that, but also shows, I think, some more non-connected. So could you tell me like what those other cells are? Because I think we see something like that in fish also.
Yeah, so those other cells, they are duct because they're all the marker positive. The problem is the technical, this is all refining again. So we used to use resin injection, kind of like Emma uses, that only gets you main trunks. It doesn't get you really fine structure. So then we went to ink. That got us even smaller particulates. We could get finer and finer structure, but we still knew we were missing part of the architecture. So now for anything that's a readout for a preclinical study, we're doing deep imaging so we can get an idea of really what is changing in the architecture. Quick assay is to inject ink, but we don't get a full picture.
Thank you. Oh, okay, go ahead.
Ken, you have a question?
Yeah, can you hear
Ken,
me?
how are you?
Fine, thank you. Thank you for sharing so exciting data with us. Very scientific question because I'm quite interested in the development. You mentioned that the left lobe was the most severe, with the most severe phenotype. Is that related to like the developmental? Do you have any like reasons for why you're seeing this? And do you see like a similar kind of phenotype in human Alagille syndromes as well, where you have a more severe phenotype in the specific lobe, for example?
Yeah, I think Dr. Piccoli can answer the second one. The first one is stay tuned. So we're doing a lot of the extrahepatic markers and following them, since we have deep imaging, following them into the liver to see how far they go in. And I'm sure, because that's your like business, Ken. So I'm sure that you probably can help us a little bit more on that, but there's a difference between lobes and contribution of how far the extrahepatic goes.
Thank you.
Dr. Piccoli.
Want to state your second question again for people?
Yeah. Ken, can you state your second?
Yeah, so Stacy mentioned that the intrahepatic duct phenotype in the JAG1 knockout was most severe, had the most severe phenotype in the left lobe in the mice. Do we kind of see kind of a similar phenotype difference in the lobes? Is there any lobe differences in the intrahepatic bile duct paucity in human patients as well?
No, so I think we believe that there is, the answer to your question is we don't know, partly because the vast majority of biopsies are done in the right lobe because we go right through the ribs and we never biopsy the left lobe. So you only have left lobes from autopsies or explants where they're so far along that I think it's probably not fair to say what is developmentally correct. I think we have been struck, at least anecdotally, that a wedge biopsy, which is predominantly subcapsular, is different than a deep needle biopsy where you're, 3 to, God help us, sometimes 5 centimeters deep into the liver when you fire that gun. And that's different. It's a different population. And probably hepatocyte mass is growing faster than ducts in that first year of life, which is why the ratio diminishes. The one thing that does go to your question, I think all pediatric hepatologists are struck by the fact that when we get big livers, we commonly have a big liver that is left lobe predominant and it sits sub-midline. And so we do a separate set of measurements, whereas in a lot of other diseases, when you have hepatomegaly, it's right lobe predominant on physical exam. But bottom line, we don't know. There is certainly regeneration in the Alagille livers seems to happen in a focal way that is kind of unique.
Thank you.
I wanted to ask Stacy a culinary question. In injecting those livers, do you ever unroof the capsule and shave just under the capsule like, you might, do with a little, peeler on a lemon or something to get, the rind, so that when you injected, you would decrease the resistance in some of the small vessels at the top so you could get to really where the action is. The hard part is all those little vessels have so much resistance and they're already filled by the time you're pushing. You have to let something blow out the top to fill them with the good stuff.
So we can try that for sure. But what we do is we clip the IVC and we clip the portal vein and we inject the bile duct first, and then we come back and inject the vessels. So in my mind, we had released pressure, but we may not have. So we can try it for sure.
Okay, any other questions? We have just a couple of minutes and then we're gonna jump off here and go to Dr. Hankinson, followed by Dr. Iwasawa. Did I do that right, Stacy?
Yes.
Okay, good. Okay.
Yes. Yes.
Any other questions? Super quick. Okay. Thank you so much, Dr. Heppert, for this, for all of your work. We are excited about Symposium and scientific meeting next year for sure. And again, if anyone is sticking around tonight, we have a special recognition for Dr. Heppert tonight at our Champion Awards. So we will see everyone in the next session in just a minute.
Thank you, everyone.
Thank you.